Welcome to my channel, My Teacher. The 16th unit in 10th biology is plant and animal hormones. In this video, I am going to explain about the plant hormones. In this lesson, we are going to discuss about the plant hormones and animal hormones. First of all, let us see the word meaning. The word hormone is derived from the Greek word hormone, meaning to excite. The function of control and coordination in plants is performed by chemical substances produced by the plants called plant hormones. In plants, several cells are capable of producing hormones. These phytohormones, that means plant hormones, are transported to different parts of the plants to perform various physiological functions. Plant hormones. Plant hormones are organic molecules that are produced at extremely low concentration in plants. These molecules control morphological, physiological and biochemical responses. Types of plant hormones. There are 5 major classes of plant hormones. They are auxins, cytokinins, gibberellins, abscisic acid, that is ABA, and ethylene. Among all these plant hormones, auxins, cytokinins and gibberellins promote plant growth, while abscisic acid and ethylene inhibit plant growth. Let us see the hormonal activities of plants. Control of cell division, cytokinin and gibberellic acid. Control of stem elongation, auxin and gibberellic acid. Initiation of flowering. Stomatal closure by abscisic acid. Development of abscission zone, ethylene and auxin. Growth of young fruits, cytokinins. Induction of fruit ripening, ethylene. Gravitropism of roots by auxin. Auxins. Auxins were the first plant hormones discovered. The term was introduced by Kogel and Hagen Smith in 1931. Auxins are produced at the tip of stems and roots from where they migrate to the zone of elongation. Charles Darwin in 1880 observed unilateral growth and curvature of canary grass, that is Phalaris canariensis coleoptiles. He came to the conclusion that some influence was transmitted from the tip of the coleoptile to the basal region. This influence was later identified as auxin by Wendt. Wendt's experiment. Fritz Walmont Wendt, a Dutch biologist, demonstrated the existence and effect of auxin in plants. He did a series of experiments in Avena coleoptiles. In his first experiment, he removed the tips of Avena coleoptiles. The cut tips did not grow, indicating that the tips produced something essential for growth. In his second experiment, he placed the agar blocks on the decapitated coleoptile tips. The coleoptile tips did not show any response. In his next experiment, he placed the detached coleoptile tips on agar blocks. After an hour, he discarded the tips and placed this agar block on the decapitated coleoptile. It grew straight up, indicating that some chemical had diffused from the cut coleoptile tips into the agar block, which stimulated the growth. From his experiments, when concluded that a chemical diffusing from the tip of coleoptiles was responsible for growth, and he named it as auxin, meaning to grow. Types of auxins. Auxins are classified into two types, namely natural auxins and synthetic auxins. First one, natural auxins. Auxins produced by the plants are called natural auxins. Example, IAA, that is indole 3 acetic acid. Second type is synthetic auxins. Artificially synthesized auxin that have properties like auxins are called as synthetic auxin. They are prepared artificially. Example, 2,4-D, that is 2,4-dichlorophenoxyacetic acid. Let us see the physiological effects of auxins. Auxins bring about a variety of physiological effects in different parts of the plant body. First one, auxins promote the elongation, that is lengthening of stems and coleoptiles, which make them to grow. Second one, auxins induce root formation at low concentration and inhibit it at higher concentration. Third one, the auxins produced by the apical buds suppress growth of lateral buds. This is called apical dominance. Fourth point, seedless fruits without fertilization are induced by the external application of auxin. This process is called parthenocarpy. Examples are watermelon, grapes, lime, etc. Fifth one, auxins prevent the formation of abscission layer. Abscission means the shedding of various parts of the plant such as leaf, fruit, flower or seed. Cytokinins. Cytokinins are the plant hormones that promote cell division by cytokinesis in plant cells. It was first isolated from herring fish sperm. Creatine was a cytokinin isolated from zea mays. Cytokinin is found abundantly in liquid endosperm of coconut. Physiological effects of cytokinin. First one, cytokinin induces cell division. That means cytokinesis in the presence of auxins. Second point, cytokinin also causes cell enlargement. Third one, both auxins and cytokinins are essential for the formation of new organs from the callus in tissue culture. That process, formation of new organs, is called morphogenesis. Fourth point, cytokinins promote the growth of lateral buds even in the presence of apical bud. Fifth point, application of cytokinin delays the process of aging in plants. This is called Richmond-Lang effect. Gibberellins. Gibberellins are the most abundantly found plant hormones. Kurosawa in 1926 observed Bacani disease or foolish seedling disease in rice crop. This internodal elongation in rice was caused by fungus Gibberella fujikuroi. The active substance was identified as gibberellic acid. Physiological effects of gibberellins. First one, application of gibberellins on plants stimulate extraordinary elongation of internode. The internodes are lengthened, so the stem length itself increases. Example, corn and pea. Second one, treatment of rosette plants with gibberellin induces sudden shoot elongation followed by flowering. This is called bolting. Gibberellins promote the production of male flowers in monoecious plant. Monoecious plants means where both male and female flowers present in the same plant. Fourth point, gibberellins break dormancy of potato tubers. Dormancy means it is a period in an organism's life cycle where growth, development and physical activity are temporarily stopped. It is broken down by the application of gibberellins. Fifth point, gibberellins are efficient than auxins in inducing the formation of seedless fruit, that is parthenocarpic fruit. What do you mean by parthenocarpy? Development of fruits without fertilization. Example, tomato. Next hormone is abscisic acid. Abscisic acid or ABA is a growth inhibitor which regulates abscission and dormancy. It increases tolerance of plants to various kinds of stress. So, it is also called as stress hormone. It is found in the chloroplast of plants. Physiological effects of abscisic acid. First one, ABA promotes the process of abscission. As we have discussed earlier, separation of leaves, flowers and fruits from the branch. It is called abscission. Second point, during water stress and drought conditions, ABA causes stomatal closure. So, it can save the plant from water loss. Third point, ABA promotes senescence in leaves by causing loss of chlorophyll. Fourth point, ABA induces bud dormancy towards the approach of winter in trees like birch. Fifth point, ABA is a powerful inhibitor of lateral bud growth in tomato. Ethylene. Ethylene is a gaseous plant hormone. It is a growth inhibitor. It is mainly concerned with maturation and ripening of fruits. Maximum synthesis of ethylene occurs during ripening of fruits like apples, bananas and melons. Physiological effects of ethylene. First one, ethylene promotes the ripening of fruits. Example, tomato, apple, mango, banana, etc. Second one, ethylene inhibits the elongation of stem and root in dicots. Third one, ethylene hastens the senescence of leaves and flowers. Fourth one, ethylene stimulates the formation of abscission zone in leaves, flowers and fruit. This leads to premature shedding. Fifth one, ethylene breaks the dormancy of buds, seeds and storage organs. Thanks for watching my video. If you like this video, kindly subscribe my channel, My Teacher.